Hello, everyone. My name is Priscilla Seabourn. I'm a APHL CDC COVID-19 Laboratory Associate, and I am working for the Hawaii Department of Health. And I'm here to discuss the Abbot Binax Now COVID-19 antigen card. So just a quick overview, the Binax Now COVID-19 antigen card, it's a lateral flow immunoassay, and it's similar to a pregnancy test. And it is used to detect the nucleocapsid protein, which is the red box over here, found on the SARS-CoV-2 virus. The great thing about the Abbot Binax Now card is that it's able to detect this protein without viral transport media. And nucleocapsid is typically detectable in nasal swabs when someone is acutely infected with COVID-19. So the principles of the procedure are similar to, like I said, that of a pregnancy test. The nasal swab is essentially mixed with extraction reagent and added to a membrane where it will move up via capillary flow. Now, the COVID-19 antigen card has been already has been embedded with SARS-CoV-2 monoclonal antibodies, and they typically will have a detector particle like a fluorophore or gold, something like that, to detect the presence of the protein. Now, the control line, which acts as an internal control for the test, should always show pink when testing samples, and that's because it, again, has already been laid down with SARS-CoV-2 monoclonal antibodies. Now, if nucleocapsid antigen is present within the nasal sample, it will bind to those monoclonal antibodies that have that, detector particle like a fluorophore, and on the sample line, it will bind, as shown here, and will show up as a pink line. Now again, the control line should always turn pink, and this is just a really good internal control to ensure that the assay is working appropriately. Now the Abbot Binax Now kit comes with antigen test cards, extraction reagents, nasal swabs, positive control swabs. And while it doesn't come with technically a specified negative control, the manufacturer recommends you use just one of the sterile nasal swabs as a sterile control. Other things that are not provided but certainly helpful is a clock or timer or a stopwatch to ensure that you have the proper incubation times. Another thing that would be helpful, it's a swab transport tube system, and this is typically probably like a sterile plastic container. And so you're going to notice that when you open the box, it comes with its own one little page manufacturer protocol. Now, we have deviated from this protocol slightly, and that's because if you follow the protocol to a T and you're one person performing the assay, when you collect the nasal swab, you're going to be put in kind of a sticky situation where you have a contaminated nasal swab in hand, and then in your other hand, you need to prep the test card, open it up, and add extraction reagent. And so unless you have another set of hands, it's going to be a little bit difficult. And so we modified the protocol slightly. However, we did get this approved by Abbot, and so they said that protocol was completely fine. And so the swab transport tubes are helpful if you're not able to test immediately, because one thing that's recommended and we'll go over is that you're not supposed to put the nasal swab once you've collected it back into its original package, and we'll talk more about that. Now, storage of the kits. So Abbot Binax recommends that you keep a kit stored between 2 and 30 degrees Celsius, which is anywhere from being refrigerated to room temperature. It's absolutely not recommended to do not freeze. It will actually inactivate some of the reagents and completely make the kit invalid after that. Now, one thing that's important is if you do decide to keep your kit in a fridge, you need to allow the kit to acclimate to room temperature before you test patients. And so I would recommend 30 to 60 minutes before testing patients to pull your kit out of the fridge and just really ensure that it's at room temperature. So an overview of what we're going to be going over. So we will discuss prepping the test card reagent, collecting the nasal swab, testing, and interpreting the results. Now again, I have modified the protocol slightly, so when you look at the card and you see that this does not match up with the manufacturer protocol, that was intentional. And so you can certainly perform the manufacturer protocol, but I would also ensure that you maybe have some help to make sure that you're not in that situation where you have to open a test card one-handed, okay? Now, performing the test. So again, something that's not provided, but highly recommended is to have the proper PPE. And so if you have access to an N95, that's great, a disposable mask, a face shield or safety glasses, disposable gloves, and some type of clothing protection, so either a disposable lab coat or a disposable gown. Now, the first thing I like to do before I do anything is just prepare my workspace. So what do you need to perform the test? And so you need a nasal swab, you need the COVID-19 extraction reagent, which is the bottle shown here, and you need a test card, okay? And again, I would also try to know beforehand if you're able to test the sample immediately, because again, you don't want to swab the patient and then, you know, you have to, and then again, you know, you're in the situation where you can't test right away. So it's good to predict that before. And so if you know you're not going to be able to test right away, make sure you have either a sterile plastic tube, label it properly, and just make sure that's available to put that nasal swab in. Again, do not put it back into the original packaging because it will actually invalidate the test. So the first thing I do is open up the test card and lay it flat on a surface. So I'm going to show you a video here where I kind of like hyperextend the card just like that, and then just lay it flat. And this is because if you don't, it tends to kind of form a V shape and it doesn't lay flat nicely like it should. So I would definitely recommend doing that to get it to lay flat. The next thing we're going to do is prepare the test card and the reagent. So hovering 1/2 inch from the top, you're going to add 6 drops of the COVID-19 reagent to the hole where the red arrow is, which is right there, okay? And again, if you're following our protocol, you need to prepare the test card within 3 minutes of before collecting the nasal swab, okay? So if you do this, you prep the card and then immediately gather the nasal swab and then immediately perform the test. So that's important to keep in mind, okay? And so here's a video showing me adding the reagent. And so see, I'm just kind of hovering right above and just adding the 6 drops to that top hole. Okay, and then just close your cap. Now, again, immediately after, you want to collect your nasal swab. And so the nasal swab, you're actually going to remove it from its package and then about 1 inch into the nostril with the most visible drainage. And obviously, if the patient doesn't have visible, drainage, just pick a nostril to start with. And then you're going to gently rotate the swab until you feel some resistance. And you're going to do this 5 times against the nasal wall, so just a circular motion. Then you're going to remove the swab and repeat the same process into the other nostril. And again, please do not return the swab to its original package because it will invalidate the results. It'll actually remove nucleocapsid antigen, if present, into that packaging. And then, if you get a negative, it could be just a false negative. And again, you want to test this within 3 minutes of preparing the test card. If you decide that beforehand, you're not able to test right away, if you place the nasal swab into a sterile plastic tube with a patient label, you can store this for up to 1 hour If you exceed 1 hour, you unfortunately need to retake the nasal swab. So just be mindful of kind of the timing of it. And here's a video. I'm actually swabbing myself just to kind of give you an idea. So yeah, about 1 inch. Okay. And when I did this, I felt some resistance, not too much. You know, it's not a nasopharyngeal swab, it's a nasal swab. So just a little bit of resistance. And then just, again, rotate 5 times against the nasal wall. And so now I'm ready to test that sample. So to perform the test, you're going to take the nasal swab, and this is a card that has already been prepared. We've already added the extraction reagent. So you're going to insert the nasal swab into the bottom hole, which is in picture A, and you're going to push upward until you see the swab in the top hole, kind of just like picture A. Then you're going to rotate 3 times clockwise or to the right, so just kind of follow that red arrow 3 times, and this allows the swab to mix with the extraction reagent, and so that you're ensuring that any antigen present is actually showing up onto the test membrane. Then you're going to remove the adhesive liner, which is shown here, and then close the card. And this is where you're going to want to have your timer or stopwatch ready and set it for 15 minutes. Now, it's important that, you know, we all are busy, but if you exceed 30 minutes before you're able to interpret the results, you unfortunately have to redo the whole process, because what can happen is if you let it set too long, it can actually self-react and start just showing positive, and you could have a false positive. So if you, even 35 minutes, you know, it's too long, I'm sorry, you're going to have to just retake it. So try to make sure you're within that 30-minute window of interpreting the results. Now, interpreting the results is pretty easy. Again, very similar to a pregnancy test. Now, the first thing I tell everyone to do is look at the control line. Okay, the control line for Abbot Binax now is made to where it should turn pink. So ensure that your control line is present and it's a nice visible pink band. Then once I ensure that the control line is there, I then look at my sample line. And so if the sample line has nothing in it, then that is a negative result. Again, if you look at the sample line and there's a pink line, then that indicates a positive result. Now, there are obviously with any type of kit, there's going to be a dud perhaps, or, you know, the potential for an internal control to fail. So this will happen when you don't see any control line, if you see only a sample line. And again, for Abbot Binax, they have made it to where their fluorophore on the control line should turn from blue to pink. If it does not turn from blue to pink, then that means that there's something wrong with the antigen card. And so even here, you know, if you see this, you might think, oh, it's positive. Well, no, because the control line did not turn pink. And so the internal controls failed, and this test needs to be repeated, unfortunately. When you're done with performing the test, all reagents and test cards are considered biohazard waste and should be thrown into the appropriate receptacle. Again, you want to use an appropriate FDA-approved disinfectant to clean up the test area. If you can actually, there's a checklist N on FDA to see if the disinfectant you are using is appropriate to kill SARS-CoV-2. And I actually have a link here on how to use that, navigate that website. Again, with any system, you know, you're going to want to ensure that the overall kit is working appropriately. And so when you receive a kit, a brand new box, it's important to perform an external positive and an external negative control. And again, I would recommend this with every brand new box or in every brand new lot number. And so the Binax Now antigen kit contains a positive control swab. And again, you can just use a sterile swab as the negative control. And this swab will monitor the entire assay. And again, I would do this with every brand new box. Now, the procedure for performing the external controls is very similar to the testing a sample patient. The biggest difference is that when you prep your test card, you're going to actually add 8 drops to that top hole, which is by the red arrow. So you do need to add a little bit more extraction reagent for the controls. And again, do not touch the card with a dropper tip while dispensing. Testing after that, like once you've prepped the test card, it's the same as testing a patient. So you're again going to insert the positive or negative control into that bottom hole, like picture A, okay? Once it's inside, you're going to rotate 3 times following the red arrow clockwise, remove the adhesive liner, close the sample card, and again, let it incubate for 15 minutes. Just the same as a patient sample card, you do not want to interpret the results after 30 minutes because they will be invalid. Interpreting the controls is very similar. Again, your negative control should be negative, but again, you want to first look at the control line, make sure that the band turned pink, then look at your sample line. So this is actually the negative control that I performed on my kit. And then my positive control looked very nice. The control line turned pink and the sample line turned pink. If your controls are failing, you have a problem. So do not use the kit if you cannot get the external controls to pass. I would put a tape on that box and just say controls failing, and then I would email Kelly Augustine. She's the Abbot technical consultant working with Hawaii right now. And I would just ping her an email and just let her know, you know, the lot number and just let her know that that external control is failing. And you can also contact myself if there's any problems. Abbot Binax also has some great additional video links. This is an active link, so you can click on it. You can see all the videos they have on Abbot Binax Now. Again, I have modified the protocol slightly just because I understand that not every facility or area is going to have 2 people to run an assay. And so I modified it so that one person could essentially run the test. So their videos are going to be slightly different from mine, but please feel free to utilize their website. They have excellent resources. If there are any additional questions, you can contact me or Dr. Joanna Kettlewell, who is also assisting with this effort. And thank you for your attention.